Cooperation, lending a hand, helping each other out. Cooperation happens in biology between all kinds of different species that can actually go out of their way to do something nice for their partner. That's not to say that both partners are always happy, though.
Complaints for the week? E. coli. Salmonella hogs up all the methionine I'm not getting enough to eat. Salmonella. E. coli takes all my hard work for granted. I work hard to produce methionine, and E. coli just poops out what I have to eat.
What Beatrice is talking about here is two grievances that our lab organisms, E. coli and Salmonella, might bring up if they were able to write. While they may seem to be in tension with each other sometimes, they're actually involved in a very special cooperation. In particular, in this flask here, they're growing together in media that neither could grow in alone.
How does this work? Well, the E. coli loves to eat lactose, a healthy source of carbon, but it lacks the machinery to make methionine, an amino acid it needs in order to survive. The Salmonella, on the other hand, can make their own methionine but cannot eat lactose, so they rely on a carbon compound secreted by the E. coli.
Although this type of reliance between E. coli and Salmonella is not found in nature, the cooperative system we constructed in lab can still teach us about the way microbes evolve together. For instance, as they evolve, will all the communities behave in the same way, or can they start to diverge from each other?
Our research leads us to believe that at least a few different interactions can occur. These plates turn blue only when E. coli and Salmonella cooperate and grow together. These photos show that there are different rates of growth between each community. E. coli and Salmonella taken from communities 1, 2, and 3 grow sooner than those taken from community 5, and community 4 has much poorer growth than all of the rest. However, at lower dilutions, where there are less of each species present, those sampled from community 5 grow sooner than the ones that were growing fastest in higher dilutions. This suggests that different mechanisms may be involved in the growth between the pairs in different communities.
We also saw differences in growth between individuals within a single community. Here are 14 different Salmonella isolates, the top 10 of which came from the same community, growing with our E. coli. As before, community growth turns the agar blue, and as you can see, some are starting to grow much faster than others. E. coli also show different patterns of growth between individuals within communities. On this plate, the sharp difference in the intensities of blue between E. coli colonies suggests that the new darker ones may have evolved a different mechanism of growth than those that are light blue. These differences in phenotype between different communities and isolates, represented by the variance in growth characteristics, leads us to believe that the genetic structure of these bacteria may have evolved in different ways.
The specific reasons for these differences can be explored by investigating the different mechanisms each community or isolate may have evolved. For example, through spent media analysis, we can determine whether one isolate of Salmonella produces more methionine than another isolate, or if one isolate of E. coli produces a better compound for Salmonella than the rest do.
The idea of spent media analysis is to pull all the cells out of media they have been growing in so that we can analyze the chemicals they have left behind. Here, we spin the cells in a centrifuge, leaving most of the cells clumped at the bottom of the tube. We can then push the remaining liquid through a filter, which traps cells in their remains but lets the chemical compounds we are interested in pass through with the rest of the liquid.
If different strains have changed their interaction in some way, we would expect them to leave behind different amounts of different chemicals in the spent media. We can test this by growing one bug in spent media from the other.
Here is a graph showing our ancestral E. coli growing on spent media from different Salmonella strains. The E. coli is growing similarly on media from Salmonella strains 1 through 4, which suggests that their interactions might be similar. But it does markedly worse on spent media from Salmonella strains 5 and 6. This suggests that communities 5 and 6 may have evolved differences in the cross-feeding interaction.
So, why do we care about this? Why should you care about this?
Microbial communities are everywhere, in the water, in the soil, even in our mouths, guts, and all over the rest of our bodies. Microbes also play key roles in industry, from manufacturing medicines to degrading waste to creating more sustainable forms of energy. And there is evidence that many of these applications could be improved by using microbial communities instead of single organisms.
For example, in a scenario of degrading waste products, one species could be specialized to perform the initial step, and the other species could be responsible for completing the process, creating an assembly line of microbial workers. So, it's worth understanding how these communities work.